Today's video is the second installment of our genetics series. Today let's start talking about Gregor Mendel, who was a 19th century monk. He lived around the year of 1850 or so over in Germany. And Gregor Mendel studied pea plants, and he looked at different characteristics of pea plants, crossed them, and made notations about the different trials that he did. And what he noticed was that there was a pattern to all of this. And that pattern brought him to a couple of realizations. Number one, there was no genetic blending. No blending. You either had a dominant or a recessive trait. You would not mix the two together. You would either be tall or you would be short. You would not be medium height. You would not blend them. And the second was that even if there is no recessive trait showing in the parents, you could still have the recessive show up in the offspring. All right, this brought him around to two laws. The first law is the law of segregation. And segregation means to split. Basically, he said that each parent is going to have two alleles, and those alleles will split when we are about to have an offspring. Split for the offspring. And this sounds a lot like going from diploid to haploid. The second law was the law of independent assortment. In the law of independent assortment, separate genes are going to be passed on independently of one another, are going to move independent. Oops, that's supposed to be a D. Independent of each other. So in other words, if I have a tall brown-haired individual, but they have the genes to be short and blonde, that the brown hair and the tall genes don't need to move together. They can go independently of one another. And I'll show this later on as we go through our crosses. But we need to know those two laws and what they mean. So let's look at a monohybrid cross. Hybrid means that we have two different alleles for the same gene. So if we're talking about height, and we said that tall was the gene which was dominant, short was the recessive gene. We could be big T, which is homozygous dominant. We could be small t, which is homozygous recessive. Or we could be a hybrid, and these are also called purebreds. All right, homozygous Or we could be a hybrid. Big T, little t, one allele of each. So this is a hybrid or heterozygous. Hetero meaning different. All right, so if we take a monohybrid cross, that means I'm taking two individuals, and we're only looking at one trait, mono meaning one trait, but we have a hybrid for that trait. So if I take two individuals that are a hybrid for that trait, so big T, small t, crossed with a big T, small t. Now, remember the law of segregation says that these alleles for this individual are going to segregate, or they are going to separate into different sex cells. So I'm going to take this guy, and my big T I'm going to put here, my small t I'm going to put here. I've segregated my alleles. I'm going to do the same thing with this individual. I'm going to segregate the alleles, big T, small t, and now we can do our cross. Big T, big T, big T, small t, big T, small t. The big T's always go, or the big letters always go in front. Little t, little t. These are our genotypes. Our genotypes tell us the alleles that we have. All right, so my genotype here is big T, or homozygous dominant. My genotype here is heterozygous or hybrid. Heterozygous or hybrid. My genotype here is homozygous recessive or purebred recessive. And so those are our genotypes. Phenotypes tell me what it looks like. So if I have two tall genes, that means I must be tall. If I have a tall and or let's go with the short genes first. If I have two short genes, then I've got no genes for becoming tall. I must be short. If I have one of each, then the dominant gene, the big T, is going to dominate over the small letter, over the recessive allele, and I'm going to have the dominant trait show up in my phenotype. So both of these would be tall. Well, let's talk about genotypic ratios. I'm going to take all the possibilities that I have. So I have the possibility of being homozygous dominant. I have the possibility of being heterozygous. And I have the possibility of being homozygous recessive. And I'm just going to count up how many of each I have. Well, I have one big T. I have two big T, small t's. And I have one small t, small t. This is my genotypic ratio for every monohybrid cross that we will ever do. We'll have a 1 to 2 to 1 ratio. Well, phenotype, remember, means what we look like, or what our individual looks like. So what possibilities would my individuals look like? Well, I could be tall here, tall, tall. So I could be tall. Or I could be short. Well, let's count them up. I have 1, 2, 3 possibilities of being tall. I have 1 possibility of being short. My phenotypic ratio is 3 to 1. Again, genotype, what are the alleles that my individuals might have? Phenotype, what are my possibilities of what I look like? And then again, remember that we showed the law of segregation when I split up my alleles. Big T, small t, we split up the alleles. All right, let's take a look at, I think all of us have done monohybrid crosses. Let's take a look at dihybrid crosses. And now I'm taking a look at 2 traits. Di meaning 2 traits. Hybrid meaning I'm going to be heterozygous for both traits. All right, so let's stick with the tall and the short. So I have tall, big T, short, little t. And P's can also be round or smooth. So we'll go big R, and we'll go, the opposite of smooth or round is wrinkled. Little r. Well, if I'm hybrid or I'm heterozygous for both traits, then I'm going to be big R, small r, big T, little t, crossed with another individual that is big R, big T, little t. And so again, we're going to have the law of segregation happen, but we're also going to have the law of independent assortment, meaning that my round genes and my tall genes are going to sort independently of one another, so I have to have all of the possibilities of these 4 letters. So I'm going to take this individual and I'm going to match them up one at a time. So I have big R, big T, big R, little t. I can take both options for this guy. And then I'm going to use this little r and go through both options for it. Little r, little t. I'm going to do the same thing with the other parent. Big R, big T, big R, little t, little r, big T, little r, little t. These 2 alleles or these 2 genes do not have to move together. So if one of my chromosomes has big R, big T, the other one has little r, little t, it does not mean that this big R and big T have to move together. It might end up being big R, little t. And you could go through that cross, and eventually we want to see the genotypic ratio and the phenotypic ratio. So pause your video right now and go ahead and do that. All right, let's see what you came up with. And I've got it figured out for you here on the next slide. And what's important to notice here, I'm not too worried. You might have made a couple of mistakes here, but I'm not too worried about these. Those will be just simple mistakes that you can always fix. What I want you to notice here are the genotypic ratios and the phenotypic ratios. The genotypic ratio, although we don't use it quite as often as the phenotypic ratio, the genotypic ratio for a dihybrid cross will always be 1 to 2 to 2 to 4 to 2 to 2 to 1 to 1 to 1. It'll always be that way. And if you were to go through and count up your genotypes here, you would see that this has happened, and I hope you did that. The phenotypic ratio for a dihybrid cross will always be 9 to 3 to 3 to 1. And if you went through and said, okay, this is going to be round and tall, all right, and you did that with all of them, you would find you'd have 9 round and tall, 3 round and short, 3 wrinkled and tall, 1 wrinkled and short possibilities. This is going to be true of all dihybrid crosses. For practice tonight, what I want you to do at home is do the crosses in numbers 1 and 3 and answer questions 2 and 4 based on those crosses. Work those out. I'll call 4 people up tomorrow in class to show us what they have and explain how they got it. So good luck on that tonight, and we'll see you tomorrow.